Okay. Remember last time I told you in the kidney, renal artery enter, correct? Renal artery being oxygenated blood, remember? See, remember previous thing a little bit. Renal artery, it bring oxygenated blood, correct? It bring oxygenated blood. But this oxygenated blood is impure. It is with nitrogenous waste. Okay, it is with nitrogenous waste. There are waste products inside it. Okay, and that waste product we need to remove, and whatever energy required for this process will be provided by, oxygen is required for the energy production. Okay, so it will bring oxygen also plus nitrogenous waste also. Okay, and renal artery bring this. When the renal artery enter into the kidney, it will divide into several arterioles. Okay, and every arteriole go towards one nephron. Correct? Remember this?
Yes, ma'am.
Now, the one arteriole, let us consider this nephron, and here one arteriole is entering. Okay, the arteriole which enter in the nephron. Okay, remember nephron is a blind sac, correct? Blind tube. It is not having opening on the proximal side. Okay, it is not having opening over here. So afferent arteriole bring oxygenated blood but impure blood into the nephron. Understanding? Now, one thing I want you to be clear with that diameter of afferent arteriole is, broad diameter. It is what? Broad diameter. Okay. And it enter into the cup of the Bowman's capsule, correct? And become glomerulus, divide into many different capillaries. And then it leave the cup shape as a efferent arteriole. The diameter is narrow over here, correct? Diameter is over here, it is narrow diameter. Okay. Now, try to understand one thing. Here, when afferent arteriole become glomerulus, glomerulus are nothing but blood capillaries. Okay. Blood capillaries. Okay. Now, remember, these blood capillaries, all blood capillaries are basically porous. Porous, you understand? With pores, with small holes. Okay, with small pores, small holes inside it. But when I talk about glomerulus, this glomerular capillaries, pore size is very large compared to normal blood vessels, compared to normal capillaries. Its pore size are 10 to 1000 times, 1000 literally, times larger. Understanding? 1000 times larger. This is one thing. Now what happened? Try to understand. The blood enter inside the glomerulus. The blood enter inside the glomerulus with very high pressure because the diameter is very high. But when it leave, very fast it enter. But when it is leaving the glomerulus, it leave very slowly. Agree? Fast entering, leaving slowly, because here diameter is larger, here diameter is smaller. Agree? So automatically, Sejal. Hello?
Nothing, ma'am.
Oh, I thought you said something.
No, I just said narrow.
Narrow, okay. Okay. So this here, they created a pressure. Agree? They created a pressure, and this pressure we called as a hydrostatic pressure. What we call? Hydrostatic. We will talk about many pressures. So remember, the pressure which is because of the broad diameter of afferent and narrow diameter of efferent, whatever pressure created on the glomerulus, it is hydrostatic pressure. Understanding? This pressure is hydrostatic pressure, and we can measure this pressure 50 mm of Hg. Understanding? This pressure is 50 mm of Hg. Remember this. And this pressure is responsible. Okay, we will talk about it later on. What this pressure do? You remember the measurement of the pressure and what this pressure is called. Okay, this is one thing. Okay, the second thing is Glomerular capillaries are very porous. A pore size is also larger. Mutti hole hai tyala, literally. It is having very large holes, literally. Okay. Understanding? Now, the next thing which you need to understand is that this Bowman's capsule, if you remember, Bowman's capsule is made up of two layers, correct? Inner layer and outer layer. Outer layer is parietal. Outer layer is parietal, inner layer is visceral. Okay, and inner layer, along with all this visceral thing, it is having those podocytes. Remember, big cells last time we studied, correct? Podocytes. Here in this diagram, they have not shown it. These podocytes are there over here, correct? I can draw only one of them. Okay, podocytes over there, and distance between the two podocytes is filtration slit. Remember these terms we did last time. Filtration slits. Okay, so there is even filtration slits. So remember these pores, there are filtration slits. Okay, this is creating pressure. Because of all these things, blood get filtrated. Blood get filtrated and filtrate enter inside over here, correct? In this space. This space is called as, what is this space called as? It is having some name. Capsular space. Okay. This space is called as capsular space. Right? So everything will come in the capsular space. Now try to understand. As porous size, see, try to understand few things. As porous size is larger, okay, pore size is larger, filtration slits are larger, lot of filtration come out, okay, lot of things come out. See, blood contains so many things. Blood contain plasma, blood contain RBC, WBC, all blood cells, okay. Blood contain lot of water, blood contain nitrogenous waste, and blood contain even nutrients, agree, glucose and whatever digested food. Everything get filtrated, literally everything enter from glomerulus, everything enter down into that pipe, into that capsular space. Only blood cells do not go down. Blood cells do not go down. There are properties of blood cells. Because of that, they do not go down into the capsular space. Blood cells remain inside capillaries. Then little amount of water will remain inside, and plasma proteins will remain inside the capillary. Everything else get filtrated. Are you understanding? Only few things remain inside blood, everything else get filtrated. Understanding? Okay. And yes, whatever remaining things, little amount of water, blood cells and plasma proteins, they will go into the efferent arteriole. Understanding? They will go into the efferent arteriole. Everything else remain inside the capsular space. Okay. Now there is a pressure responsible for it. We will talk about it. But I want you to see what happened to efferent arteriole. Okay. See in this our previous diagram. Okay. If you see over here, this efferent arteriole, okay, is coming down, correct, and becoming blood capillaries which surround the renal tubule, correct? These are called as peritubular capillaries. All of them together called as peritubular capillaries. Understanding? Peritubular capillaries. See, they surround over here and they are now called as peritubular capillaries. Okay. So whatever little thing remains, go and surround the renal tubule. Understanding? Because whatever blood want also, whatever blood need also, that is also taken by renal tubule. Correct? Understanding? See, blood need whatever digested food. Right now it is in the renal tubule. Okay. Blood need almost all the water is taken by renal tubule. Correct? Even blood unwanted things as well as required things, everything is taken by renal tubule. Now this efferent arteriole need to collect things back, correct? Otherwise, suppose it has not
Renal
collected in
tubule and renal capsule is different.
capsular space. This space is capsular space. Capsular space. Renal capsule is not the thing. Capsular space is this space, and this is renal tubule from here to here. Okay.
Okay, ma'am.
Okay, and renal capsule is no renal capsule. No, there is nothing
In
called renal
small
capsule.
Malphizian body. Malphizian body is renal capsule. Corpus, sorry. Sorry.
Got it? No, there is nothing called a renal capsule. Okay. I don't know anything called renal capsule. Or maybe like, see, there are terms. I told
It
you.
was corpuscle, sorry.
Okay. Yeah. So this thing, this peritubular capillary. See, in biology there are hell lot of terms. Okay. In all subjects, I think I teach biology, so I realize in the biology, but in all subjects, और biology के कुछ ज़्यादा ही है.
No, ma'am. You know, in physics, like we denote pressure by P. So there are thousands of units
P.
and whatever we define by P. It
That's very,
confuses.
very difficult. Physics is very difficult and it confuses till last. And solving problem in physics is difficult. See, understanding physics is okay. You can solve theorems and all. Okay. Even whatever kids like are preparing this year for NEET, no, they were telling the same thing. Board physics is okay. You can do it, theorem and all. But solving problem when it actually come in NEET, it's really, really difficult. Okay. And you need to solve it in very pressurized situation, exam pressure time. So better to practice it a lot. Kids are telling, all kids tell, if they solved a similar problem, only values are different, then only they able to solve in exam. Otherwise no, if it is new problem, no one able to solve in exam. Okay, so practice more and more. Physics is like, oh, hell difficult. Okay, let's come back to the point. So remember, renal tubule, whatever renal tubule contain, okay, that will become urine. Understanding? What will remain inside the renal tubule, that will become urine. Are you understanding? Sejal and Prachi,
Yes, ma'am.
understanding? So whatever blood need to take it back, correct? Otherwise, everything our body will throw out, correct? That's why that next process is called as the selective reabsorption. Selective reabsorption. So after ultrafiltration, like Bowman's capsule is ready to throw everything out, okay? But blood is not ready for that. So blood go around, collect the things which is required, select the things which it requires, and that process is called a selective reabsorption. Understanding?
Yes, ma'am.
We will move
Yes,
across.
ma'am.
Yeah, we will move to the selective reabsorption. But before that, we need to understand a little factor in the ultrafiltration. Okay, I told you what happened in the ultrafiltration, but I did not tell you what is responsible for ultrafiltration. So let us talk about what thing is responsible for ultrafiltration. I'm removing this stuff. So yeah, I want you to remember the hydrostatic pressure. Hydrostatic pressure is let us write it. Okay. Now this whole ultrafiltration process is carried out by the pressure gradient. It is filtration. Filtration is always carried by pressure gradient. Correct? Pressure is responsible for filtration always. Okay.
We even
So
have hydrostatic pressure in physics.
yeah, of course.
That fluid is at rest.
And in this one topic you study in 11th, it is biomolecule topic. That same topic is there in chemistry, 12th standard chemistry.
Yes, ma'am.
Oh, yes, ma'am.
Sir told us.
Same topic, exactly same topic. Okay. And same biomolecule topic, half of the topic this year you will learn in the molecular biology. Okay. Half of the topic, not whole thing, half of the topic. So it is repetition. If you could relate the things, it will become easy. Okay, it will become easy for you. Yeah, so remember the pressure which is responsible, I'll just rewrite it. Pressure which is responsible for ultrafiltration, okay? Pressure responsible for ultrafiltration process, it is called ultrafiltration is called as effective filtration pressure. Effective filtration pressure, EFP. Now EFP we can calculate, okay? EFP, effective filtration pressure, is equal to hydrostatic pressure. Hydrostatic pressure. Now you know what is hydrostatic pressure. Hydrostatic pressure is created due to the larger diameter of afferent and smaller diameter of efferent, correct? Now we know even value of it, 50 mm of He. We will write it little later. Now remember, this hydrostatic pressure causes filtration. Agree? It is a positive pressure. It causes filtration. Agree? But there are other pressure which are, which opposes filtration. They don't want filtration to take place. Understanding? So effective filtration pressure is positive pressure minus negative pressure. So let us talk about what are the negative pressures. Okay. The first negative pressure is basically both the negative pressures are osmotic pressure. Let me tell you which those pressures are. 1 second. Now, one thing is that blood capillary, when I talk about glomerular capillaries, they contain many substances, correct? They contain many substances, like they contain plasma proteins, there are cells, okay? Plasma proteins plus cells and several other substances, which really don't want to go out of capillaries, agree? Which really don't want to go out of capillaries.
Yes,
see, any substance don't want to change its state, correct? So these substances also don't want to go out, and they opposes filtration, and that filtration is called as osmotic pressure. That pressure is called as osmotic pressure. Remember, osmotic pressure opposes filtration. Opposes filtration. That's why it is a negative pressure. Okay. It is a negative pressure and it is 30 mm of Hg. There is one more hydrostatic pressure. See, now see, this is 30 mm of Hg, but this is 50, right? So what happened? Still filtration is taking place, okay? And filtrate is coming into this space, correct? Capsular space. Now this filtrate is called as nephric filtrate. Remember this, okay? Now this filtrate is called as nephric filtrate, or even called as glomerular filtrate. Any one name you can write. Glomerular filtrate. Okay. Now when I talk about glomerular filtrate, here it get accumulated, correct? Here it is a lot. That's why here it get accumulated. And when here fluid is accumulated, it don't want any more fluid to come in, correct? It don't want any more fluid to come in. It will opposes filtration. Fine. It will also opposes filtration. And this pressure which opposes filtration, which is created by nephric filtrate, it is called as the filtrate hydrostatic pressure. Filtrate hydrostatic pressure. If you see note, you will find all these things, okay? Hydrostatic pressure, and it is 15 mm of Hg. So remember, two negative pressures, 130, 115, correct? So this become 45, and here we are having, oh, this pressure is 55, positive pressure is 55 and this is 45. So 10 mm of Hg. This is a net pressure which causes filtration. Understanding? Got it?
Yes, ma'am.
Yes, ma'am.
So 2 negative, 1 positive, 1 is 55, not 50. I told you 50, but it is basically 55. Okay. So this is about filtration. Fine. Now, let's go
Yes.
to the next point. Let's go to the next point. Okay. Now, selective reabsorption. Selective reabsorption is very, very simple. I will just tell you and you will understand. Peritubular capillaries roam around or wrap around, literally wrap around the renal tubule. Renal tubule, this pipe is carrying unwanted things, literally unwanted things. But blood want to select some things from it. Okay, so it will do selective reabsorption. Okay, now remember the renal tubule structures. One is PCT, proximal convoluted tubule, then loop of Henle, then distal convoluted tubule. Okay, it is very simple. So I'll simply tell you. Now, everything blood has, important things also are there in the renal tubule. Blood need to take them back. Okay, so blood use two mechanisms for it. One is passive transport, that is just by osmosis the things will get transferred. Okay, from renal tubule to blood capillaries. Do you understand osmosis? Osmosis, process of osmosis.
Ma'am, in 8th standard it was there, I think,
At
of
11th also there.
raisins.
11th also there.
That experiment we had.
Cell topic, osmosis. In the cell topic, the most important thing you will learn is osmosis. Okay, understand it very, very well. Okay, but right now I'm not going in detail of osmosis. You prepare it in the cell topic. You are having a, like in the plasma membrane, you have osmosis in depth. And in 11th topic, I don't know in state board thing. Just a second. Huh? There is one topic called transport in plants. You have it in 12th or 11th? In CBSE, it is in 11th. Last year, it was deleted. So in that osmosis, there is a lot of osmosis. Transport in plant. You had that topic last year?
No, I think.
Just a second. Huh? Plant-water relationship. You have this year. Okay, in that topic also there is a lot of osmosis. Means transport in plant is totally dependent on osmosis because plant don't have heart, no? Okay, we have heart for transport. Okay, plant-water relationship means transport in plant. Like that is, you know, what is it? Let me tell you in short. See, just imagine a very tall tree. Okay, imagine a coconut tree. Okay, such a large tree, correct? But a coconut tree also, water will be available only to roots, and leaves need water. So that plant need to pump that water from root to leaves, correct? At least 30 feet coconut trees. Have you seen coconut tree?
Yes, ma'am.
Yes, ma'am.
Okay, so that pumping mechanism is basically depend on osmosis. Okay. So osmosis, anyhow, you are going to learn this year. So I'm not telling you what is osmosis. It is a wastage of time now. So remember, the two methods are applied to take the substances by blood, okay, by blood capillaries. One is passive transport, which is by osmosis, substances just enter. And remember, passive transport means, always remember this, word passive means no energy, okay. Passive means no energy. Automatically the things will happen. It is called as a passively happening. Okay. So passive transport do not need energy. Understanding? It do not use energy. But all substances are not transported without energy, correct? Because see, renal tubule have its membrane. Understanding? Blood capillaries have its membrane. So substances need to cross two membranes, right? Substances need to cross two membranes and go. Everything cannot go without energy, two membranes crossing, correct? So many substances are transported from renal tubule to peritubular capillaries. When I say the word, try to recollect it. Renal tubule is our, where the vest is there, okay? And peritubular capillaries are our blood capillaries. Okay, so just 1 minute is remaining. I'm disconnecting this call and I'll share another link. Okay, it will take 2 minutes because I like put on recording. Okay, I'll share another link. Okay, otherwise it will disconnect in between. Okay.